ensure that the new facility is fit for the purpose and that the design of the new facility qualification status are appropriate to accommodate the process to be transferred. There should be this gap assessment, and risk assessment should include resources, including the human resources, to ensure that the personnel on the receiving unit having the adequate qualification, the adequate training, the adequate process understanding of the received technology, and that the responsibilities between the sending unit and receiving unit are well described and well documented. A documentation of all the technology transfer activity is fundamental to ensure that gap assessment, training plans, plan for technology transfer, risk assessment conducted are all well documented and kept, and accordingly, process sample verification testing is conducted. And additionally, logistical consideration for the international transfer of the product that will be transferred, either bulk or starting material or drug substance. Another important challenge or limitation is the use of CPV versus APR. Some manufacturers interpret that continuous process verification and annual product is a redundancy, and we repeat trending of the product. And then it's worth mentioning that both continuous process verification and annual product quality reviews are both regulatory requirements. And although they are similar in the objective, that both are maintaining the state of control and identifying continuous improvement initiative, yet each one have its characteristic and its unique purpose. So in the continuous process verification, there is a timely response to process trends or signal. So it allows reaching trends and identifying signals in the real time. It's maximally efficient, agile, flexible pharmaceutical manufacturing. So it allows manufacturer to ensure the capability and process performance prior to releasing the batch to the market. And it focuses more on the critical process parameter and critical material attributes. While the annual product quality review is a high-level summary of product performance, but it involves several inputs that are not included or considered in the continuous process verifications, like complaints, regulatory submissions, and so on, stability behavior, and so on. And it's minimally performed annually. So it doesn't give the real-time response that is achieved by implementing continuous process verification. They complement each other. You may integrate them in some sort, so you may use the trends, some of the trends established in the continuous process verification while formulating and preparing your annual product review, but each one is a regulatory requirement and each one have its unique objective. Process validation, the life cycle approach we talked about, is focusing on prospective validation. And we all know that there are other types of process validation that is well known. And this classification of the different types of process validation depends on the time of performing the validation in relevance to the manufacturing and marketing decision. So in the prospective validation, product will be manufactured with a new formula or within a new facility. It's a pre-market validation. The product is not yet commercially distributed, and it's carried out before commencing routine production. It's the foundational type. It's the type of choice for the validation because it is the starting point for any product that will be released under new condition. And in this type, we build confidence in product quality before allowing and releasing the product to the market. Another type is retrospective validation, and this is conducted only when the manufacturing process has not formally undergone a documented validation. It uses historical data and trend analysis to provide evidence that the process is in a state that it is intended to be, but it is no longer an acceptable approach for process validation because it is not acceptable that the product to be validated after its commercial distribution. The concurrent validation, which is conducted during regular pharmaceutical production to demonstrate that the process performs at a level that it should be in the course of its actual execution, it's rarely acceptable approach to process validation under specific circumstances where the risk of implementing this concurrent can be achieved for criticality or critical demand of the process. But it should be used rarely and should be pre-approved from the regulating authority. And finally, the revalidation, which is not widely used for drug products. It was following the traditional approach when the prospective validation reached a conclusion that manufacturing process is unable to produce the product consistently, and criteria for revalidation indicated in the original validation protocol, but now with the current process validation approach, revalidation is no longer used because if we see any signals or any trends in the continuous process verification, we go back to stage 2 in the process qualification, or we go further in the process design. So no longer revalidation activity, we have the continuous process verification. If everything is in a state of control and the process capability is maintained, so the routine monitoring continue. If there is any abnormal trend or any need to review the process according to the encountered risk, according to the identified gaps, we should go back to stage 2 or stage 1 to adjust and modify. Finally, how we present this control strategy in the Common Technical Document dossier. A clear presentation of the control strategy and its development is essential to realize the added value of enhanced knowledge in the marketing authorization. It doesn't have a dedicated part in the dossier. It should be spread across the dossier. For drug substance, part S2 is the most suitable for summary of drug substance control strategy element, and for drug product, similarly, P2 is the most suitable for summary of the drug product control. And by this, we conclude the presentation and we will finish by the case study. The first case study is defining process performance qualification batches, CPP and IPC. Here is an example of hepatitis B, recombinant hepatitis B vaccine. When we perform the prospective validation of it, and what is the appropriate approach to define the critical in-process testing and critical process parameters while we are planning the control strategy. So the most appropriate approach is to go step by step to define what are the critical in-process control and what are the critical process parameters. For example, in the seed, the optical density is very important, and the morphology culture that have characteristics of the cell, and there is no evidence of other contamination present is very important. And after that, we go for the steps for the fermentation. During the fermentation, the antigen production is done by propagation of the cells and antigen expression. So in the fermentation, to ensure the increase in the count of the cells, we measure the optical density, and we verify also the morphology of the culture, that it should be, each cell should be round shape and budding with no any sign of external contamination. And methanol in this step is used for the antigen expression. So the optimal concentration of the methanol is critical to is a critical process parameter, and hence, as long as the methanol concentration is critical, one of the critical process parameters and one of the critical in process control should be the methanol estimation by the gas chromatography. After that, it is the start of the separation activities, when we separate by performing cell washing and centrifugation. In centrifugation, the rotation speed is critical process parameter that may impact the final antigen concentration and antigenicity. So the speed, RPM, is very important, and the conductivity of the supernatant is also should be checked. After separation, there is the cell lysis step, where we verify optical density, feed flow rate, temperature. Contact time is very important. So one of the most important CPPs is the inlet temperature, the outlet temperature, and the antigen test using Akon test. After that, the polyethylene glycol precipitation and clarification to start the purification of the bulk. So in this step, incubation time with the polyethylene glycol per hours and the Akon test to ensure the antigen. Another purification step is the aerosol washing, which is reliant on the adsorption time. And as you know, the adsorption and desorption process is significantly influenced by the temperature, the contact time, the pH. So while washing and contact with the aerosol, the adsorption time is the critical process parameter, and verifying that the aerosol wash is free from any antigen using the Akon test is important in process control. Next step is the aerosol desorption by filtration. And the desorption is influenced by temperature. So the temperature is a critical process parameter, and the circulation time is another. And definitely the in-process test will remain the antigen content. Then purification by ion exchange, which requires size exclusion. And here we ensure that the elution of the low molecular weight, 0.2 molecular weight, pH conductivity, and antigen test is positive, and that this low molecular weight is containing the antigen, and high molecular weight elution, pH and conductivity, and antigen test showing negative test. Then tangential flow for concentration, tangential flow filtration system. And as a filtration system, the pressure is the main critical parameter. So we measure the inlet pressure, we measure the pressure across the membrane, and the in-process test is the antigen test to ensure that there is no loss on the yield. Ultracentrifugation, the parameters, the speed, the temperature, the filtered antigen, including all the intermediate